All right, let's jump right in. Today, we're talking about a huge challenge in modern medicine. What happens when a precision cancer drug, one of these miracle therapies, just stops working? We're going to crack the GIST code. So think about it like this. You have a groundbreaking drug that's like a perfect key. It's designed to fit a very specific lock on a cancer cell and shut the whole thing down. It's an idea that has totally changed the game for patients. But the mystery we're digging into today is a really tough one. What happens when the cancer is smart enough to change the locks? Our story focuses on a cancer called a gastrointestinal stromal tumor, or GIST for short. It's the most common sarcoma you find in the GI tract, and for a long time, it's been the absolute poster child for this new era of targeted cancer therapy. And here's why. For most GISTs, the problem boils down to something very specific. You've got these two genes, KIT and PDGFRA, that get a mutation, and it's like their on switch gets jammed. They just keep telling the cell to grow and divide, completely out of control. So these are the locks we're talking about. And for those specific locks, science came up with the perfect key, a drug called imatinib. I mean, this thing was a game changer. It fits right into those broken proteins, turns them off, and stops the cancer in its tracks. The results were so incredible, it became the textbook case for precision medicine. But here's where the perfect story starts to get a little complicated. You see, while most GISTs have those KIT or PDGFRA mutations, a pretty big chunk, about 10 to 15% of them, don't. These are called wild-type GISTs, and they don't have the usual locks for our key to fit into. And that leads to the million-dollar question, right? If they don't have those standard mutations, what on earth is making them grow? And maybe more importantly, why are they so often resistant to imatinib, our supposedly perfect key? This is where the real detective work had to start. Now, to solve this kind of mystery, researchers needed a much more powerful magnifying glass. So they turned to something called next-generation sequencing, or NGS. Just think of it as a super fast scanner that can read a tumor's entire genetic blueprint, start to finish, looking for any and all hidden clues in the DNA. And this technology is really the heart of a whole new genetic detectives toolkit. Not only can they use NGS to scan the tumor itself, but they can also do something amazing called a liquid biopsy. This is where they find tiny fragments of tumor DNA just floating around in a patient's blood. It lets them monitor the tumor in real time, watching how it changes and evolves, all from a simple blood draw. It's incredible. So as they started looking deeper, they had to separate two different kinds of genetic chaos. The first is what's called tumor mutational burden, or TMB. That's just the total number of mutations. The second is chromosomal instability, or CIN. That's way bigger. It's about errors in the whole structure, like huge chunks of DNA getting copied or deleted. It's kind of the difference between a few typos in a book and having entire pages ripped out. And the clues started coming in fast. First, they found that, surprisingly, most GISTs have a pretty low number of individual mutations, a low TMB. But, and this is a big but, the really aggressive drug-resistant tumors often had high chromosomal instability. That high CIN was a major red flag for a bad outcome. And, most importantly, it was very often linked to a mutation in one very important gene. To really understand the stakes, we have to remember this isn't just about data in a lab. This quote from a research paper brings it all home. It's about a real patient, a real person whose cancer didn't respond to the best drug they had. Solving this mystery was literally a matter of life and death. And these scans, wow, they show you just how ferocious these resistant cancers can be. On the left, you've got the tumor at the beginning. On the right, only a few months later, you can see it has just exploded, spreading into the liver. This thing is not a slow burn. It is a wildfire. So, what was the secret ingredient driving all of this? After digging into the patient's tumor DNA, the detectives found their culprit. It wasn't in KIT or PDGFRA. The cancer hadn't just changed the locks. It had disabled the entire building security system. The problem was a hidden mutation in a gene called TP53. Now, TP53 is not just any gene. It's so important it has a nickname, the guardian of the genome. Its whole job is to be the cell's ultimate quality control manager. If a cell's DNA gets damaged, TP53 steps in and either makes it pause for repairs or tells the cell to self-destruct. But when TP53 itself is mutated and broken, that guardian is off duty, and damaged cells are free to multiply, accumulate more errors, and run wild.
And here's the smoking gun. Using those liquid biopsies, researchers tracked this culprit over time. In the original tumor, the TP53 mutation was super rare, just 0.1% of cells. But watch what happens. As the cancer fights off treatment, that number climbs. 6 months later, it's at 2.7% in the blood. A year later, it's nearly 10%. And by the time they tested the cancer that had spread, that one TP53 mutation was in 99% of the cancer cells. The cells with the broken guardian had completely taken over the whole operation.
The researchers' conclusion was just crystal clear. They called it an indisputable association, a direct link between these dangerous TP53 mutations and an aggressive type of GIST that is completely insensitive to imatinib. The hidden enemy was finally out in the open.
So what does this all mean? Well, it means the old battle plan has to be completely rewritten. It's not enough to just check for KIT and PDGFRA anymore. You've got to profile deeper and find the true driver. Once you know what's really going on, you can tailor the therapy and use tools like liquid biopsies to stay one step ahead of the cancer as it tries to evolve. And that really is the future. That's the whole point. The goal is to get away from a one-size-fits-all approach and move towards something truly personalized. It's about reading the unique genetic story of each person's tumor to find the best way to fight it.
And why does this matter so much? Because this isn't just theory. It means we can stop giving patients treatments that we know won't work. It helps doctors spot the most dangerous tumors right from the very beginning. And sometimes it can even point to a totally different drug that might work. In some of these TP53 cases, a drug called sunitinib actually showed some promise. You're turning the tumor's own genetic code into a roadmap for how to defeat it. So it really leaves us with this one big, hopeful question. We now have the power to read a tumor's entire genetic story, to understand its tricks and find its weaknesses. With that kind of power in our hands, how long will it really be until the whole idea of one-size-fits-all cancer treatment is just a relic of the past?